Hello, everyone. I am really glad to have you here today as we start a very key topic in clinical chemistry, water and electrolyte balance. So this might seem like a basic concept, but it really play a central role in how the body functions, and even small imbalance can lead to serious health issues. So at the end of this chapter, I hope that we all better understand how the body maintains fluid and electrolyte level. We understand the role of body water in relation to electrolyte balance. List four principal serum electrolyte and their functions, and also reference ranges. Remember, these reference ranges are not going to be given to you during AXCP exam, so hopefully you might be able to memorize them. Calculate the ion gap using the electrolyte result, and also understand the causes of abnormal electrolytes and ion gap. Define osmolality and examine the relationship between serum osmolality and the urine osmolality and serum electrolyte concentration. We also discuss the measure and calculated osmolality, then calculate the osmolality and osmolar gap using the laboratory result and correlate result with clinical symptoms. Okay, so next we're able to examine how hormones like ADH, aldosterone, epinephrine, norepinephrine regulate water and electrolyte, especially in response to changes in osmolality and blood volume. As you know, the hormonal balance is critical in maintaining homeostasis under stress, right? Then we able to describe the some of the metabolite, which is categorized, how they are broken into measurable compounds like nephrine and VMA. We compare and contrast the primary hypertension with hypertension caused by excessive amount of catecholamide, then list the laboratory test used in different differential diagnosis of various type of hypertension. Eventually, you're able to um, understand the clinical symptoms and lab findings in conditions such as pheochromocytoma, SA88, symptom of inappropriate of ADH secretion, diabetes insipidus, and dehydration. We understand how serotonin is synthesized and degraded in the body, and eventually discuss the clinical value of measuring 5-HIAA, a key serotonin metabolite in diagnosis and monitoring. All right, so let's start our very first slide with body water. So water is a solvent for all purposes in human body. This means the water serves as the medium where all biochemical reactions occur, right? So the functions of water, we have transports nutrition to the cells. Water acts like a body delivery system, carrying dissolved nutrients through the bloodstream to every cells. So for example, glucose from food dissolves in water and travel through blood vessel to reach cells that need energy, right? We also have the functions of water of determining cell volumes. Water moves in and out cells through osmosis, as you already heard of it, directly controlling cell size. So the right amount of water maintains proper cell shape and function. So yeah, you already heard about it. I'm sure that too much water causes cells to swell and too little cause them to shrink. Then another function of water is to remove waste products by way of urine. So the kidney filter metabolic waste from blood and dissolves them in water and create urine. This include, for example, nitrogen waste like urea and excessive salt, for example. We also have the function of water is to act as a body coolant by the way of sweating. Then the intricate balance exists between body water and electrolyte, which is the electrolyte is defined as constituents of cells and ions capable of carrying electrical charge. So too much water dilute electrolyte dangerously. So if the patient actually had the blood draw contaminated with IV fluid, so what happened to the electrolyte is going to be falsely low, right? Because too much water dilute electrolytes, while too little water makes electrolyte concentrated too high. So your test result will be falsely high. Total body water can divide it into two major compartments. We call it intracellular fluid and extracellular fluid. So intracellular fluid predominantly potassium. This is the water inside our cells, intracellular fluid, which makes about 60% of total body water. Potassium maintain the predominant component here because cells membrane actively pump potassium inside while keep sodium out. And that's why when the blood is being hemolyzed, you all know that it has a high potassium concentration that leaked into the plasma or a serum. Extracellular fluid um, is the water outside of the cells, predominance sodium and chloride, including the plasma and the fluid between the cells as well. Water molecules can move randomly across the permeable membranes, and normal plasma is usually 93% of water. Remaining is the lipid and protein. So blood plasma composition does show how the water dominant our circulatory system is, right? So the remain 7%, including all of these component protein, um, albumins, the clotting factors, hormones, lipoproteins, transport the cholesterol, all of that. We also have to mention that the this high water content allow plasma effectively dissolve and transport nutrients, waste products, and also gases throughout the body. I really like this chart as it does show the composition of body fluid chart composition. So the ionic composition of body fluid. So this chart does show us the different concentration of electrolyte and other charged particle in three main fluid compartments of the body, measured in millimole per liter. So first of all would be the plasma component. The plasma component, as you already been aware, is sodiums, including chloride, that dominate about 140 millimole per liter each, making them the primary electrolyte in blood. So bicarbonate is present about 20 millimole per liter and serves as our main buffer system to maintain the pH. You also have a really little amount of potassium, calcium, and magnesium here. The second one would be the interstitial fluid composition. So this fluid between the cells has almost identical electrolyte concentration to plasma because they actually just separated by capillary wall that freely almost uh, allow ions to pass through. The main differences here is that interstitial fluid contain fewer protein since most plasma protein are too large to cross capillary wall easily. The one that make a little different here is intracellular fluid. So it does show us a dramatically different pattern. Potassium dominates about 140 um, millimole per liter here, and while sodium is only very low at about 10 millimole per liter. So, as you know, this reversal from extra fluid is maintained by the sodium potassium pump. You also have phosphate compounds and protein serves as the main anion inside the cells, replacing chloride, which is most excluded here. You have a chloride here in plasma, right? This is another way of looking at our water, sodium, potassium distribution. So water and sodium, potassium are the key substance that are distributed across the body fluid compartments. The intracellular fluid, which is around 40% of body weight, is about containing potassium at 1 to 10 millimole per liter and sodium as only 10 millimole per liter. Extracellular fluid is 20%. As the water is outside of the cells, split between plasma and interstitial fluid, this extracellular compartment has the opposite pattern. Again, sodium as 140 millimole per liter and potassium is only at 4. You also have a very active transport. For example, you have a sodium out, 2 potassium in, maintained by sodium potassium pump in the cell membrane. This pump works constantly to maintain the concentration gradients against the natural tendency for these ions to equilibrate. You also have the passive movement here versus the active transport. The sodium and potassium naturally want to move down to their concentration gradients. So, for example, if the sodium wants to leak into cells and potassium want to leak out, this the active transport pump must work continuously to counter this passive movement. So, in general, just understand that you also have another transportation system beside the active transport using the pump, using the cell membrane movement with the ATP involved. You also have passive movement here. So the system entirely maintain the electrical potential differences across the cell membranes, which is essential for nerve and muscle function. You do want to maintain the electrical neutrality among all of those components outside or inside the cells. So the total plasma solute concentration here, the cation and anion. The bottom line here is the total cations plus anions is around 308 millimole per liter. This represents the total osmotic activities of the plasma, which determine how water moves across membranes and help maintain proper blood volume and pressures. So let's break it down. The cations total is 154. The anion also 154 for both. So this demonstrates the electrical neutrality, which we just mentioned, right? So this is the reference ranges that most of us are responsible for. The sodium reference range is 136 to 145. And remember, all of these electrolyte reference ranges can vary slightly depending on the laboratory because we use a different instrumentations and the methodologies. So sodium is a primary extracellular cations and the main determinant for plasma osmolality. The potassium is reference is about 3.5 to 5.1. So they are mainly intercellular. Plasma level must be tightly controlled during this range. As you know, if it's a little bit greater than this, you call it critical for the high or low potassiums, right? Chloride is the major extracellular anion that follows sodium to maintain electrical neutrality. So if the reference ranges is around 98 to 108 millimole per liter. Lastly, we have carbon dioxide. The reference ranges is around 23 to 29 millimole per liter. So, as you might already been aware, when you run the carbon dioxide to report, this actually measure bicarbonate, which is our primary buffer system to maintain the blood pH. So we run bicarbonate and report as the carbon dioxide because it does reflect the body ability to balance acid base. So these four electrolytes together provide crucial information about kidney function, fluid status, and also acid-base balance. Now we are going to embark into a very first electrolyte component, which is sodium. It plays a major role in maintaining osmotic pressure due to its high concentration in extracellular fluids. So the sodium is the most abundant ions outside of the cells. Remember, it's about 140 millimole per liter. It is the primary determinant on how body water stay in the extracellular space. It has the vital function in membrane potential, for example, muscle contraction and nerve conduction. Sodium is also the major counter ion that available when metabolically produced acids becomes neutralized to produce their conjugated base. So what does this mean? When the body produces acids, for example, during metabolism, right? A lot of metabolism functions produce acid. These acids might be buffered to prevent the pH changes. Sodium serves as a positive ions that pairs with negatively charged conjugated base form when acids are neutralized. So these allow these buffer compound to be safely transported and eliminate. For example, what you can see here is the lactic acid. So the lactic acid essentially being replaced by sodium in this equations. So sodium combined with the lactate ions, while bicarbonate neutralize the hydrogen ions, forming sodium lactate, which can be safely metabolized, and carbonic acid, which essentially later converted into s- uh, CO2 and water that can be exhaled through the lungs. So that's an example of how our body prevents the dangerous acidosis that would occur if lactic acid accumulate unbuffered. Sodium level in body fluids are maintained by the renal reabsorptions and proximal convoluted tubules based on the sodium threshold, which is controlled by sodium potassium ATPase pump. The sodium balance is also maintained by the influence of hormones. For example, the aldosterone on the distal convoluted tubules as a consequence of the changes in blood volume and pressures. So the kidneys has two level control system for sodium here. The first is the proximal tubules reabsorb about 65% of filtered sodium using the sodium potassium pump. Secondary, you also have aldosterone, which is the hormones that is released when your blood pressure drop or blood volume decrease. Aldosterone is released as a result to increase sodium reabsorption, which retain water and restore blood volume and pressure. When we have extremely high or low sodium concentrations in plasma, will cause severe osmotic pressure changes that can induce serious consequences to several organs. So, since the sodium is the primary determinant of plasma osmolality, abnormal sodium level, whether it's high or low, create a dangerous water shifts. So, for example, the low sodium cause water to move into the cells by osmosis leading to what? Cellular swelling. So this particularly dangerous, right? Because the our brain cannot accommodate the swelling. High sodium hypernatremia cause opposite effect. The cells shrink as water leaves them. Then what happened to this? It lead to the swelling on the brain, leading to potential coma and seriously death. All right, so that is what we have just talked about, but in the extended level. Delusional effect. So loss of water, diarrhea, sweating, and burns causes loss of water. So when the body lose water without proportional sodium loss, the remaining sodium become concentrated, right? So it leading to hypernatremia. So diarrhea cause water loss from the intestine, excessive sweating, loss of hypertonic fluid. We also losing more water than sodium. Burns, for example. Retention of water is causes by renal failure, congestive heart failure. So when the body retain the excessive water without proportional sodium retention, what happened is that we have hyponatremia. The existing sodium become diluted. In renal failure, the kidney cannot properly excrete water. That's the basic function of kidney, right? In congestive heart failure, we have poor circulations that trigger hormones that retain both water and sodium, but often more water is retained, diluting the sodium concentration. Osmolality and sodium level can help to differentiate renal loss of water versus non-renal causes. So that is the job of the clinician have to figure it out. Hyponatremia or hypernatremia. So in hyponatremia, low sodium with high osmolality, it usually may be due to hyperglycemia. So when blood glucose is extremely high, it's created osmotic pressure that's pull water out of the cells into bloodstream. This dilute the sodium concentration even through the total body sodium may be normal. The high glucose maintain the high osmolality despite low sodium. So this is called pseudo hyponatremia because it's not due to the sodium itself because it's high glucose. Lastly, we have hypernatremia, high sodium with low urine osmolality, maybe due to diabetes insipidus. So in diabetes insipidus, the body cannot concentrate urine properly due to the ADH hormone excessively, I mean, lack of ADH hormone, not excessively, lack of ADH hormone or kidney resistance to ADH. So no matter what happened, whether the lack of ADH hormones or kidney resistance, it caused massive water loss through the dilution of urine while retaining sodium, leading to high blood sodium, but actually paradoxically low urine concentration. Next, we have potassium, as we already learned about it. Is a major intercellular cation that is controlled by the sodium potassium ATPase pump. So while most potassium stay inside the cells at the concentration around 140 millimole per liter, the sodium potassium pump actively maintain um, the distribution of this potassium, and that's why it stays in very narrow range. Potassium maintains the cardiac rhythm and contributes to the neuromuscular conduction. And any imbalances in potassium level, as indicated by potassium hyperkalemia and hypokalemia, low sodium, low potassium, will cause cardiac arrhythmias and neuromuscular weakness. So, for example, in our nerves and skeletal muscle, potassium help establish the resting membrane potential and contribute to the electrical signal that needed for muscle contraction. We have an abnormal potassium level, whether it's higher or low. So when we have a hyperkalemia, it could be due to the decreased renal function, such as a chronic renal failure. If a kidney fail, it cannot effectively excrete potassium through urine. A response to electrolyte imbalance, such as acidosis. So during acidosis, the excessive hydrogen ions enter the cells to be buffered and potassium ions actually exit the cell in exchange to maintain electrolyte neutrality. So that's why it had raising potassium level, even the total body potassium may be normal. Then artifactually, such as sample hemolysis, as you already know, it falsely elevated due to the the blood cells breaking down during the sample collection and processing hemolysis. Now we have a hypokalemia, which is due to GI loss, such as vomiting and diarrhea. The GI tract normally reabsorbs more dietary potassium, but the chronic diarrhea, for example, is particularly problematic because intestinal secretions contain high potassium concentration. Renal loss, such as hyperaldosteronism. So excessive amount of aldosterone hormones cause the kidneys to retain sodium while wasting potassium. So we also have the another reasons for hypokalemia because of the response to electrolyte imbalance, such as alkalosis. So in alkalosis, the body attempts to normalize pH by moving hydrogen ions outside of the cells, and potassium ions move into the cells to replace them. And that's why you have the cellular shift due to the plasma potassium concentration. All right, next we have chloride. So chloride is one of the major extracellular anion that help maintain electrical neutrality with sodium. The ingested diet in diet and almost completely absorbed by intestinal tract, as chloride comes mainly from salt, dietary salt, and is efficiently absorbed by the small intestine along with sodium. This chlorides actually have a little bit more complex absorption mechanism. So the chloride absorption is nearly one efficient, meaning almost all dietary cholesterol enter the bloodstream. Chloride enter the bloodstream. So this is something that really important in acid-base balance. Chloride contributes to the maintenance of acid-base balance by participating in isohydric shift, which is exchange for the bicarbonate and chloride shift. So what does it mean? In the red blood cells, as CO2 is picked up in tissues, it forms bicarbonate inside the cells. To maintain the electrical neutrality, the bicarbonate exits the red cells while the chloride enters the cells. So this is a chloride shift or isohydric shift. So this mechanism helps transport CO2 from tissue to the lungs while maintaining proper ionic balance. Hypokalemia may follow hypokalemia may follow hyponatremia or may be caused by loss of chloride due to such condition as vomiting. So what does this mean? The low chloride typically accompanies low sodium in dilutional states. So, for example, when chloride can be lost during the vomiting process, which remove hydrochloric acid, because this is predominantly in our stomach, right? So, hydrochloric acid from stomach is also being removed, leading to hypochloremic metabolic alkalosis as the body compensates for acid loss. So I really like this mm, diagram as well, as it does explain us this principle. This is the transport and buffering of carbon dioxide and red cells, erythrocytes. So the chloride ship is illustrated by the chloride, by the is the by the CO2 transportations that maintaining the ionic balance in red cells as they carry CO2 from tissues to the lung. This is what happened. In tissues, the CO2 entered the cells, entered the red cells, and combined with water through the help of carbonic anhydrase, which is the enzyme to form the carbonic acid. H2CO3. This immediately dissociates into hydrogen and bicarbonate. So what happened is the hydrogen ions are buffered by hemoglobin, preventing the dangerous pH changes. The bicarbonate exits the the red cells into plasma, and to maintain the electrical neutrality, the chloride enter the cell. So that's why we call this as chloride shift. Then in the lung, the process reverse. The bicarbonate, remember, now you can ask the questions. So now the red cells now have a lot more of a bicarbonate, but the process reverse. The bicarbonate re-enter the red cell from the plasma while the chloride exits, right? So inside the cells, bicarbonate combine with hydrogen ion. And then what happened next? Reform carbonic acid, which then break down to CO2 and water. The CO2 essentially diffused out the blood and exhaled during the respiratory process. All right, next we have bicarbonate. Bicarbonate is the major buffer for metabolically produced acid. So the bicarbonate buffer system is our primary defense against acid buildup in normal metabolism. It reabsorbs in the proximal convoluted tubules and um, distal convoluted tubules. Venous, serum, plasma, CO2 versus bicarbonate. So in clinical laboratory tests, CO2 is actually measured the total CO2 content. Again, very important, primarily bicarbonate by 95% plus small amount of dissolved CO2 and carbonic acid. Decreased bicarbonate may associate with the conditions such as metabolic acidosis. So when bicarbonate level drop, it indicates the body has used up the buffer, reverse trying to neutralize excessive acid, right? So increased bicarbonate may be associated with the conditions such as metabolic alkalosis. So it's often result from the loss of stomach acid, as we already talked about through vomiting, for example. So let's take a look in the four principal plasma electrolyte reference ranges because we are in the lab, we use these reference ranges to calculate anion gap. So the ion gap can be calculated by two different equations. The reference ranges for ion gap is 10 to 20 if we use the anion gap equation with potassium in here. The reference ranges for the one without the potassium is 7 to 14 millimole per liter. When electrolytes are in balance, there are about 10 to 20 more anions than cation in plasma. So these anions include phosphate, sulfate, protein, organic acids such as lactate, citrate, pyruvate, acetic acid, 3-hydroxybutyrate constitute the anion gap, which is the concentration of unmeasured anions. So all of these unmeasured anions are normal components that plasma contributes. Um, but not usually measured in routine chemistry panel, for example, CMP. So the plasma proteins are particularly significant contributors because they carry multiple negative charge as a physiological pH. Most of the unmeasured anions are conjugated base of metabolic acid. So when the body produces acids during the normal and or abnormal metabolism, these acids again are buffered by bicarbonate, right? So when the lactic, for example, if the patient comes in and you measure the lactic acid, is really high, the lactic acid become lactate and keto acid become keto anions. In the conditions such as lactic acidosis, diabetes, ketoacidosis, for example, which is very poisonous in the body system. The causes of increased anion gap. So when you calculate, and usually the machine calculated for you, if you see an increased anion gap, what does it mean? It means it's metabolic acidosis actually occurred. So this is the explanation. Increased production of acid leading to decreased amount of bicarbonate in plasma, leading to the increase of ion gap. So, for example, when you have the DKA, diabetic ketoacidosis, the lack of insulin causes the cells to break down fat for energy, producing the ketoacidosis. So, ketoacidosis, remember we have acetoacetic acid, beta hydroxybutyric acid. So, these acids are buffered by carbonate, forming keto anions, which is acetoacetate and beta hydroxytubate that are not usually measured in routine electrolyte panel, and that's why the result is low bicarbonate and high anion gap. We have another example is hypoxia. So when you have hypoxia, when tissues don't receive adequate amount of oxygen, they switch to anaerobic metabolism. So producing excessive amount of lactic acid, and this lactic acid is buffered by bicarbonate, creating the lactate anions that contribute to the anion gap. Common causes include shock, severe anemia, for example. Renal failure is due to the increased retention of normal acid and leading to decrease of bicarbonate. So usually in healthy kidneys, excrete various organic acids produced during metabolism. In kidney failure, these acids accumulate because they cannot eliminate effectively. So abnormal concentration of sodium. That is the one of the causes of increase in anion gap. So in renal disease, is not reabsorbed from PCT back into the plasma. When the kidney are damaged, they lose the ability to reabsorb sodium efficiently uh, from the proximal tubules. We also have the abnormal secretion of aldosterone, leading to the abnormal reabsorption from the distal convoluted tubules back into plasma. So aldosterone, as you know, the aldosterone-related disorders can affect sodium reabsorption in distal tubules. The increase of potassium does not cause the increase anion gap because it's not compatible with life. So at this time, the patient is really in dangerous situation. Severe high potassium usually cause fatal cardiac shock before it can significantly affect the anion gap calculation. So it's really life-threatening hyperkalemia condition. Chloride usually does not change the ion gap because it crosses the cell membrane easily and never accumulated or get used up in the reaction. Um, Unlike the bicarbonate, it gets consumed during the acid buffering, and that's why chloride level remain relatively stable and move passively to maintain the electrolyte neutrality. Remember that chloride shift. So now we are going to switch gears a little bit and discuss the importance of osmosis, osmotic pressure, and oncotic pressures. So these are important the pressures existing in our body to regulate water. Osmosis is a diffusion of a liquid, most often water, through a semi-permeable membranes from a low solute concentration to high solute concentration. So the osmosis is the natural movement of water across the membranes to maintain the concentration of the solutes on both sides. So the water usually move from the fewer dissolved particle. So lower solute concentration to the more dissolved particles, which is the higher solute concentration to maintain the equilibrium. The presence of more solute on one side of the membrane generate the pressures known as osmotic pressure. So osmotic pressures is the force that drive water movement across the membrane. The size of higher solute concentration pulls water towards it, creating a measurable pressure. The osmotic pressure generated plasma protein is known as colloidal osmotic or oncotic pressure. So this is a little different here because plasma proteins is involved in this process, such as albumin. They are large molecules that cannot cross capillary wall easily, so they remain in the blood vessels. These proteins create a specific type of osmotic pressures called oncotic pressure or easier refer to oncotic pressure or colloidal osmotic pressures. So these oncotic pressure is low, like in protein malnutrition, when the oncotic pressure is low, when the patient has a protein malnutrition, the fluid accumulates in tissues causing edema, and that's the reason why sometimes you see the patient swelling as a result of the, for example, the protein malnutrition. So this is a short and simple diagram that actually helps us explain what does this mean to have the balance of oncotic and hydrostatic pressure along the capillary. So hydrostatic pressure, for example, 32, 27, 18, and 15. So this is how it start high at the arterial end and gradually decrease as blood flows towards the venous end. The oncotic pressure is constant at 24. Uh, the oncotic pressure created by plasma protein, again, it remained constant at 24 throughout the capillary because of what? Protein cannot cross the capillary wall, right? So this pressure actually pulled the fluid back into capillary from the tissue space. Now, you have the protein concentration difference create the oncotic pressure gradient. For example, high protein concentration inside the capillary, for example, 0.8 versus low protein concentration in the interstitial fluid generate the 24 oncotic pressure that retain the fluid circulation. So again, you are not required to memorize or remember these number, but this chart just really show you the proper fluid exchange and the importance of the oncotic pressure with involved in the protein and how it's regulated water filtration from at the arterial end and reabsorption at venous end and how our body constantly maintain the tissue hydration while preventing edema. Osmolality measure the number of dissolved particles, for example, the molecules of ions in the solution, is usually expressed in milliosmoles per kilogram of water. Sodium and chloride are major contributor to serum osmolality because they are major particle of extracellular fluid. Glucose and urea also contribute. Normal plasma osmolality is usually 275 to 300, and osmolality of urine a little bit different from 300 to 900. The way we calculated the osmolality is using these expanded or simplified. So for the purpose of this course, we are going to use this one. 2 sodium plus glucose over 20 plus BUN divided by 3. So this version is easier to remember and calculate it quickly, though we also remember that ASCP can ask you to calculate using this formula. All right. So no matter what formulas that are utilized is help estimate the plasma osmolality when direct measurement is not available. So this is a quick estimation. So why do we have to use this calculation? Remember, the calculated osmolality should closely match with the measured osmolality. the measure is the one that used by the machines, right? If let's say if there is a significant difference between the osmolar gap based on the one that being measured or based on the one that being calculated, that mean it suggests the presence of unmeasured um, substances. For example, what happens if the patient coming in with high alcohol, methanols, or other toxins? That is the reason why we have this calculation that make the calculated osmolality as and is considered as a very useful tool in detecting poisoning or any other metabolic disorder. So in the lab, you might already been seen the osmometer. So the osmometer is utilizing the um, principle of cooling the sample below its freezing point. They initiate the crystallizations and measure the exact temperature at which freezing occurred. So modern osmometer are highly accurate and detect the differences as a very 1 to 2 um, MOSM per kilogram. So it is really accurate. Osmolar gap. So osmolar gap can detect the measure abnormal concentration of osmotically active solutes. So that means we already consider sodium, urea, glucose by comparing the calculated osmolality with measured osmolality. So the way we calculate it is to have measured osmolality from the instrument minus the calculated osmolality. Then you have the osmolar gap. Usually osmolar gap is stated as 9 mol/sm per kilogram, representing what? The contribution of unmeasured normal plasma components such as protein, potassium, organic acids. This small gap is already accounted for in the calculated osmolality formula. So when the gap exceeds 10 to 15 mol/sm per kilogram, it's suggest abnormal substances that are present. So give you an example here. If the patient coming in the ER and suspected with DUI, this is the formula that we are going to be looking at. This example, the elevated gap of 25 millimole per liter times to the 40 milligram per mole shows that the house osmolar gap can quantify toxins level. So you have the suspected ethanol poisoning. The measure from the instrument is 320. Then you use the formula to calculate osmolality. Then you have 220 minus 295. Then you have a 25. This 25 does suggest that the elevated gap, so the elevated gap of 25 indicates of ethanol present. So if you suspect the ethanol present, then you use the molecular weight of ethanol time to the 25 and compared to the DUI or DWI level, if greater than 80, and this person is way more than 80. So that's this calculation is also work for any other active toxin. Um, Other than ethanol, even though ethanol is the one that usually very prevalent in forensic sciences and in law enforcement. Clinical significance of osmolality. So, first of all, is increased osmolality due to sensation of thirst, which is a very natural response that our body produce. So, when plasma osmolality rise above normal, which is way greater than 295, right? Specialized in the brain. So the brain actually detect this change and trigger a sensation of thirst. So we have that sensation of thirst, prompting us to drink more water to dilute the blood and restore normal osmolality. Increased water consumption diluted the extracellular fluid and decreased the osmolality. So if you keep drinking water, in response to thirst. Add water into the extracellular fluid without adding solute. What happened? This dilute concentration of dissolved particle, effectively lowering plasma osmolality back toward normal. The water distribute throughout the body compartments according to the osmotic gradients, restoring proper fluid balance. You also have the secretion of ADH of vasopressin. They are secreted by posterior pituitary gland and act on the kidneys to increase water reabsorption. Once osmolality decreases, the ADH secretion is turned off. You have major hormones that control the body blood volume, the ADH, the antidiuretic hormones or vasopressin from posterior pituitary gland, and aldosterone for cortex of an adrenal gland. You also have two major hormones that regulate blood pressure. So that is the blood volume. This is the blood pressure. You have epinephrine and norepinephrine. Epinephrine is from the adrenal medulla and norepinephrine from adrenal medulla. Mechanism of the ADH. So the two major of normal hormones control the body blood volume, right? ADH. So first of all, ADH is the major determinant of renal water excretion. It's synthesized in the hypothalamus and stored in posterior pituitary. It's the stimulus happen is when the blood become more concentrated. What happened is the osmoreceptor in hypothalamus near the posterior pituitary detect this change. These specialized cells can sense even small increase in osmolality and trigger ADH release. Increased plasma osmolality. Rising plasma osmolality above, let's say, 295 is the primary stimulus for ADH secretion. The action is to travel to the collecting ducts of kidneys and leading to more water reabsorptions. Once released, the ADH travel through the bloodstream to the kidneys, where it's binding to the receptor, so that it can um, produce more of the action of water reabsorption. Mechanism of aldosterone. So aldosterone secretion is regulated by renin angiotensin system. Its stimulus is due to the cells from the JGA glomerulus apparatus in nephron arterial sends low blood flow to the kidney. What happens is when you have a low blood volume, low blood pressure, it's all stimulates the JGA cells detect it and initiate the renin-angiotensin cascade. So this is how it works. JGA detected uh, low secretion, a low amount of sodium, and the blood volume being low is initiate the renin. Angios and renin initiate the renin acts like an enzyme that split the angiotensinogen to form angiotensin 1. Angiotensin 1 initiate angiotensin 2, leading to vasoconstriction, which is the vasoconstriction of blood pressure. And in providing what happened is we have the rapid blood pressure. So now angiotensin 3 is occur leading to the adrenal cortex, and then you have aldosterone. So now you have the longer term volume regulation component of the system. So at this time, because of all of this cascade, the aldosterone action on the kidney tubule leading to the sodium reabsorption, the aldosterone travel to the kidney where it binds to the receptor of distal convoluted tubules and collecting ducts. It increase the channels and sodium potassium pump, increase the sodium reabsorption in the urine. So that is the verbatims of what we have just talked about. In short, renin is a highly specific protease enzyme that cleaves in the single peptide bond in and angiotensinogen. Then you have the angiotensinogen, which is a alpha 2 protein synthesized in the liver. Uh, the plasma carry the angiotensin 1 to lung, where protease enzyme, angiotensin converting enzyme, ACE, converted into the angiotensin 2. Then angiotensin 2 has two actions: vasoconstriction, which increase blood volume, and then circulated through the plasma to the adrenal cortex that stimulate the secretion of aldosterone. This provides the long-term volume regulations that complements its immediate pressure effect. Another mechanism of aldosterone. So aldosterone circulated through the plasma to kidneys, where it stimulates distal convoluted tubules to reabsorb sodium and also associate water molecule. Because when you reabsorb sodium, it's also reabsorb water molecule the from the future back into plasma, eventually increase the blood volume. Aldosterone also stimulate the potassium excretion. As aldosterone increase in sodium reabsorption, it simultaneously enhance potassium secretion into the urine. So that's why you have the net result as increase the blood volume and increase the blood pressure. So this is a chart that I would like to put it here as hopefully it will give you a brief explanation of of how the links between the water and sodium metabolism. All right, so another one that we are going to dissect here is the mechanism of epinephrine and norepinephrine. So the epinephrine and norepinephrines are called catecholamine due to their chemical structure. Is epinephrines is usually in predominant size. It constitutes 90% of all catecholamines produced. The stimulus is the nerve impulse from sympathetic nervous system due to the physiological and psychological threat. Its action is enhance the heart muscle uh, irritability of heart muscle, then increase the heart rate and increase the strength of each heartbeat, then essentially increase the blood pressure. You have the primary hypertension. The primary hypertension is known as essential or idiopathic hypertension. That is essential, meaning is net doesn't mean it's necessary or beneficial. Idiopathic meaning of unknown origin. So it's cause unknown and treatment is symptomatic since we cannot treat underlying cause because it's unknown. Treatment is focused on the lowering blood pressure including the 95% of case of hypertension. But at this point, usually no abnormal lab test is a key diagnostic feature. There's another condition here. This is actually a rare tumor of adrenal medulla, pheochromocytoma, which is a rare neuroendocrine tumor that develops in the adrenal medulla. It caused by increased secretion of epinephrine and norepinephrine, leading to increased blood pressure. It's usually cause increased production of metabolized metanephrine and normetanephrine, leading to increase of metabolic components is called vanillyl malic acid, VMA. So the lab test that usually measure the VMA is the lab test is usually measuring the VMA, epinephrine, and no epinephrine in the urine. So the diagnosis heavily relies on measuring the elevated level of these catecholamines and their metabolites in 24-hour urine collections. Aldosterone-induced hypertension. So another tumor that actually produced leading to the excessive production of aldosterone. So as you know, when your aldosterone is increased, you have increased blood volume, leading to the increased blood pressure. Then at this time, you will measure the urinary excretion of aldosterone and sodium and potassium. Um, so one of the last one that we will cover is the syndrome of inappropriate ADH due to tumor producing excessive ADH. So SIADH occurs when ADH is secreted inappropriately, meaning is released when it shouldn't be, typically from ectopic source like lung tumors, brain tumors, or certain medication. So if our body is healthy, we usually have ADH regulations, right? So unlike the normal ADH regulations, where release depends on osmolality and volume status, the source of produce the sources produce ADH continuously regardless of the body actual water. So it doesn't care if our body uh, actual water body water and balance actually in checked. Um, Increased blood pressure leading to increased blood volume, increase in the blood pressure. So the excessive ADH causes kidney to reabsorb too much water from the urine, leading to the water retention and blood volume expansion. It's really very difficult to measure ADH by lab test, and measuring serum and urine osmolality is one of the keys measurement here. Excessive ADH leading to inappropriate dilute serum and concentrated urine. The excess ADH can cause excessive water reabsorption by the kidney, leading to the water intoxication at this point. The next one is the diabetes insipidus. So it's a little bit different than the SIADH. Over here, you have the excessive amount of ADH increase. Now you have the lack of ADH secretion or lack of a kidney response to the ADH. A huge volume of urine is excreted. Then you have dehydration. Your body telling you to drink more water because of sensation of thirst, right? So now you keep drinking fluid, keep drinking water, more urine production. Then you essentially have a decreased urine sodium. It does not cause abnormal blood pressure because other mechanisms. So at this time, you have epinephrine and aldosterone compensate. So patients with diabetes insipidus typically maintain normal body blood pressure through the several compensatory mechanism. So remember we talked about the renin-angiotensin aldosterone system is actually activated to retain sodium and stimulate thirst. Last one is serotonin. It is a hormone produced by intestinal tumor resulting in the episodic hypertension. So in order for us to understand this hormone, this um, episodic hypertension, we usually measure the 5-hydroxyl indoacetic acid, 5-HIAA, and the specimen of choice is your urine.